Hello, everybody. Thank you and welcome to this special I3 session, which brings you the highlights of the 13th Biennial Congress of Alpha Scientists in Reproductive Medicine, which was held just recently in September in Seville, Spain. We had an enormously successful Congress and a lot of fun, right? Everybody would second that. And this session, if you missed the Congress, will be the next best thing, minus, of course, the fantastic food and drinks that we all enjoyed in Seville. We encourage you to participate in this session and ask questions, but please do so using the Q&A section of the platform and not the chat function. One very important notice, please stay to the end of the session as we will be announcing the location of Alpha 2024 Congress, and you will have a chance to win, actually two chances to win free registration to that conference and 1 year of free membership in Alpha. Which then brings you some nice benefits, which I will tell you about later on in the meeting. So it is now my pleasure to introduce my co-moderators and co-hosts. I'm very happy to be here with all of them. They're all known to you. I will give you a brief bio, but their full bios are available on the I3 website. And I encourage you to go to the website and learn more about these wonderful people. So Dr. Alison Campbell is the Chief Scientific Officer at Care Fertility. Alison has over 25 years of experience in the field of reproductive medicine and has played a key role in establishing and integrating new laboratories, driving standards and best practice, and leading scientific research and development across the Care group. Dr. Sharon Mortimer has been a clinical embryologist since 1985, and over the years has been a consultant in ART, developing, designing, and improving art programs. She has been a CFAS certified art laboratory scientific director since 2013. And just recently, and congratulations to you, Sharon, became a certified organizational coach. And of course, we all need that. we all need coaching in that area. And of course, Sharon is the immediate past president of Alpha. And I should mention that Alison is the current vice president of Alpha. Thank you both for being here. Then we have Dr. Dean Morbeck. All of you know Dean. He's currently a freelance consultant based in the US and New Zealand. Lucky you, Dean. And he's an adjunct associate professor of obstetrics and gynecology at Monash University. In addition to his extensive experience as a lab director, Dean has been involved in clinical and translational research focused on quality in the laboratory, from culture media, protein and oil, and the use of KPIs to the promising integration of artificial intelligence. Then last but not least, we have Professor Christophe Blockeel. Christophe is a medical director of the Center for Reproductive Medicine at University Hospital Brussels in Belgium. He has worked in the field of fertility for several years, and his primary interest is reproductive endocrinology. He has published around 100 papers in peer-reviewed journals and lectured widely at numerous international meetings. We are very lucky to have Christophe as a member of the executive committee of Alpha. Thank you so much. I'm going to now pass it on to Alison.
Thank you very much. Thank you, Mina. It's really a pleasure to be here this evening. I've been so excited about this event all day, and it's taken me back to Seville in the summer with the sunshine and the yoga and the great talks. So really, I'll just introduce you, if that's okay, Mina, first.
Sure.
Really needs no introduction. Dr. Mina Alikani, so our president of the Alpha Society, clinical embryologist, certified as a high complexity clinical lab director and a clinical consultant by the American Board of Bioanalysis. So lots of letters after your name, PhD from Monash University, and very long career, very experienced, and we're all learning from you every day. Lots of publications, about more than 80 articles and book chapters on laboratory aspects of assisted reproduction. So a huge contribution to the field. So that's Mina, and we've already been introduced to Sharon Mortimer. So it is with great pleasure that I introduced Dr. Sharon Mortimer's lecture talk this evening, which will be the, in case you missed it, Seville 2022. Thanks very much, Sharon.
Thank you, Alison. Thank you for the opportunity to speak again and to revisit that wonderful conference. And thank you, everybody, for joining us. So I'm just going to quickly go over, give you a quick overview of the conference for those who missed it, and a journey down memory lane for those who are there. So first of all, we thank our sponsors and exhibitors for their support of the conference. And we also like to thank Asabir for their cooperation and contributions to the Alpha program and the conference that were very instrumental in at least one of the sessions. So this is the current Alpha Executive Committee, and since David Gardner's term finished during the pandemic, we didn't get a chance to thank him in person. So again, we'd like to thank him now for all that all the contributions that he made to Alpha during his term as the president. We also want to introduce Yasmin. She is the Secretariat for Alpha, and so she's the person that you'll talk to if and when you contact Alpha. So Alpha, where did we come from? Well, in the beginning, Alpha was founded in 1994 by Jacques Cohen and Brian Dale, Steen Villetsen and Yves Menezo. Professor Edwards was the honorary president, and the aim of Alpha was to further human reproductive medicine in all aspects, to bring together embryologists from all fields to promote international understanding, cooperation, and collaboration. Another aspect of where did we come from is remembering those who've gone before us and whose work and discoveries that we rely on every day. So we honour all of those pioneers who've passed away since the last Alpha meeting in 2018, including those who are represented here. And what have we done as Alpha? Well, in the past, before the internet, we published newsletters from 1995 to 2008. We had innovations like skill-based workshops, groundbreaking internet conferences that were managed by Kay Elder and Thomas Elliott in the 1990s. And that was the first time that we'd actually been able to do something online that involved everybody across the world. They were 24-hour conferences, and they resulted in the publication of these books. Alpha was also associated with some of the early master's programs around the world in clinical embryology. And of course, everyone knows Alpha for the consensus meetings. And these citations and reads data just show how much influence those publications have had on our daily practice, and these results on the citations and reads are from today. There was also an international agreement on the knowledge and skills that one needs to have to be able to call oneself a clinical embryologist. And this was done in association with ESHRE, ASRM and Aspire. And we worked together to develop frameworks for required knowledge, for skills at all the different levels, as well as trying to harmonise position titles, roles and responsibilities. And of course, the Alpha conferences. These are a great opportunity for embryologists to meet, learn and socialise, and they've been legendary ever since the first one that was held in Sorrento, Italy back in 1997. So let's go to Spain. So here we are meeting at the conference in October. The opportunity for learning. There were lots of opportunities for learning, both inside the hall and outside. I'm sure you can't read everything on this slide. It's quite small. I had to squeeze it up to get the program in. But the parts you can read, you can see that we covered a great range of topics, from cutting edge new basic research through to some translational research and practice tips that people could use immediately. Here's some more opportunities for learning from each other inside and outside the hall, and then lots of opportunities for networking and interaction as well. So today we're delighted to be able to bring you the highlights of the presentations from the Alpha's 13th Biennial Conference. We have 2 prize papers and a paper that has a huge impact and influence on our daily practice. So I'm looking forward to being able to present those all to you, and we're looking forward to hearing the speakers. So with that, I will leave you. I'll just say thank you. Thank you to those who joined us in Seville, and thank you to those who are joining us today.
Great. Thank you, Sharon, for quite a review of really, a field that we're fortunate to have had leaders with the foresight to put together Alpha almost 30 years ago. I'd like to do a call out though for Thomas Elliott. You mentioned him. He was doing those internet sessions in the 90s. Most of you on this call maybe weren't even out of, maybe some out of diapers, but Thomas Elliott was really, way ahead of his time putting those together. Those were some amazing sessions. So I'd like to next go to our first speaker for today, and it's really a pleasure to introduce Dr. Alessandra Alteri. Dr. Alteri is an ESTRA-certified senior embryologist who obtained her PhD in cell and developmental biology at Sapienza at the University of Rome, and she started her career as an embryologist in Rome, and since 2015 has been working at IRCCS San Raffaele Scientific Institute in Milan. In 2017, she was elected junior deputy of the Special Interest Group for Safety and Quality at ESTRA. And she's also been an adjunct professor of biology at Vita Salute San Raffaele, University of Milan. And she's going to tell us whether we should hatch or not on frozen, thawed, or warmed blastocysts. So, welcome.
Thank you, Dean, for this introduction. So, good evening or good afternoon, depending on where you are. First of all, I would like to thank Alpha because this is my first award of my life, of my career. And performing a randomized control trial is a huge effort, not only for embryologists, but also for clinicians. But it's so important to do it. So this is an award for all the staff of my IVF clinics. So thank you, Alpha. Okay, regarding our study, so as we know, in vivo, an essential step for successful implantation is the escape of blastocyst from the zona pellucida. At the blastocyst stage, the embryo is subjected to a phenomenon called hatching, which includes the expansion of the blastocyst with stretching, thinning, rupture of the zona, and finally the extrusion of the blastocyst through the zona. But prolonged exposure of blastocyst of human blastocyst in artificial conditions is thought to impair their ability to implant. In fact, in vitro, hatching process and also zona pellucida are altered due to zona hardening following culture and cryopreservation, zona abnormalities following ovarian stimulation and fertilization, and finally the entrapment of the blastocyst in the zona. The artificial rupture of the zona pellucida is known as assisted hatching, and it is used in order to improve the chance for implantation during assisted reproduction. But NICE guidelines do not recommend the use of assisted hatching because it has not been shown to improve pregnancy rates. Moreover, assisted hatching is considered as an add-on treatment by HFEA. Very recently, in addition, the American Society for Reproductive Medicine declared that assisted hatching should not be recommended for all patients because there is insufficient evidence that assisted hatching could benefit also specific groups of patients, for example, patients with poor prognosis. But most of the confusion surrounding this topic is probably due to the fact that assisted hatching procedures highly vary in terms of time and mode of application. And these procedures could have completely different consequences on hatching dynamics. And this leads to a strong heterogeneity among the studies. In fact, we can perform assisted hatching using laser currently, but also in the past by mechanical cutting or chemical solution. We can perform assisted hatching on the 3, 5, 6, 7 of embryo development in fresh and frozen cycles in different groups of patients, advanced maternal age or recurrent implantation failure. And finally, we can perform assisted hatching, making holes or breaching or thinning of different sizes. So many studies focused on the effect of assisted hatching are in cleavage stage embryos in fresh and frozen cycles. But on the other hand, few data regarding the effect of assisted hatching are available for blastocysts, especially after cryopreservation. In fact, 2 randomized control trials and few observational studies addressed clinical outcomes derived from partial hatched blastocysts in frozen cycles. Although some benefits emerged from these studies, the heterogeneity did not permit to draw any conclusions regarding the real effect of assisted hatching. So, in order to contribute to fill this gap in the literature, we performed a randomized control trial in order to understand whether a partial zona pellucida removal by assisted hatching of vitrified-warmed blastocysts can improve clinical outcomes. The Aladdin study is a multicentric prospective comparative study with a parallel randomized control design, which includes 2 infertility centers in Milan. All women undergoing non-donor cycles and scheduled for a single embryo transfer were enrolled. And we included the women aged under 40, undergoing first or second frozen cycle using vitrified blastocysts, and less than 2 previous ovocell retrievals. We excluded the PGT cycles, BMI more than 35, abnormal uterine cavity, and finally severe male factor. Our primary outcome was live birth rate, and we did the sample size analysis in order to achieve 80% of power to rule out a 10% difference in live birth rate between assisted hatching and non-assisted hatching groups. So we required 350 blastocysts per arm, overall 700. Regarding secondary outcomes, we considered biochemical and clinical pregnancy, miscarriage, multiple pregnancy, obstetric, and neonatal complication rates. The analysis was performed including cycles between September 2018 and November 2021. And we randomized 698 blastocysts, 352 in the assisted hatching group and 346 in the control group. Let's move on to the protocol. Blastocysts were vitrified once expansion has been reached on day 5, 6, or 7, and after artificial shrinkage. The randomization was performed immediately after warming, and assisted hatching was carried out using laser pulses from 1 to 5, 6 o'clock position of the blastocyst in order to remove 1/3 of the visualized surface of the zona. The blastocyst survival was evaluated 1 hour after warming, and blastocyst was transferred at least 2 hours post-warming. So let's see our results. As expected, both groups show similar baseline patients' characteristics. As for baseline frozen embryo transfer characteristics, 2 groups show similarities. But regarding clinical outcomes, no significant difference was found in terms of the primary outcome, live birth rate, and also clinical pregnancy and miscarriage rate between assisted hatching and control group. Concerning obstetrical and neonatal complication, no relevant difference was observed, confirming that this treatment is a safe procedure. But the randomization was performed stratifying by age over and under 38, and considering 2 populations once again, no difference has been observed between assisted hatching and control groups in terms of live birth rate, clinical pregnancy rate, and also miscarriage rate. Finally, we perform exploratory subgroup analysis for the primary outcome in order to understand whether assisted hatching could have some benefit in a specific situation. And as you can see, we failed to identify any subgroup that could benefit from assisted hatching. So our trial is a multicentric trial with a large sample size, but it shows some limitations. First of all, the heterogeneity in clinical practice between 2 centers may represent a major confounding factor in interpreting the results. Secondly, in this study, it is not possible to blind the researchers, embryologists, to the group allocation of patients. And finally, at this stage, it is not possible to evaluate long-term outcomes of children. So we can conclude that no significant difference has been identified between assisted hatching and control group in terms of live birth rate, also stratifying by age. But so assisted hatching on vitrified-warmed blastocysts seems not to improve clinical outcomes, but further studies may be required to rule out milder, potentially interesting benefits of assisted hatching on vitrified-warmed blastocysts in terms of live birth in subgroup patients. So thank you for your attention.
Very nice. Congratulations, Alessandra. This was amazing. A very nice RCT. Congratulations with your work. I am allowed to ask you one question for now, because as you know, I'm a reproductive endocrinologist. So I would like to ask you something with regards to the endocrinologically, let's say the hormone levels during ovarian stimulation. Do you think it might influence the blastocyst behavior, as we can call it, and implantation potential after assisted hatching? What do you think about so estradiol levels, progesterone levels, and so on? Could it have an influence?
Thank you, Christophe, for this very interesting question. I know that hormone levels could have an impact on blastocyst, blastocyst dynamics. In our study, no difference was found in terms of hormone levels between 2 groups. But as you can see, our population is characterized by good prognosis patients.
Exactly.
So it would, I think that it would be very interesting to investigate the role of the effect of assisted hatching also in other population, for example, poor prognosis patients, or also after several previous failure. But in our study, no difference was observed.
Okay. Very nice. I think there is more questions, and there is already more questions in the chat, but this is for the Q&A session. So thank you again, Alessandra, for your nice talk. And this bridges very well to the next speaker, ladies and gentlemen, who is Ainhoa Larreategui, if I pronounce well. She's a biochemist, graduated at the Autonomous University of Barcelona in Spain, and she is specialized in ART at the University of Nottingham. She is now a third-year doctoral student under guidance of Professor Stout and Professor Kops at the Utrecht University in the Netherlands. So she will tell us everything about the centrosome failure and the peri-centrosome fragmentation in the first cell division of the non-rodent mammalian zygotes. So please, Ainhoa, the floor is yours.
Many thanks, Christophe, and many thanks for Alpha for giving me this opportunity to present again this project, which I think it's amazing, and it's a great opportunity also for, I think, the people who couldn't attend the Congress to see it now. So I will just start with my presentation, and I always like to start with the fact, because it's an idea of what worries our society and a way to address a problem. And here the problem would be that about 10 to 20% of known pregnancies, they end up in a miscarriage. And this number can be even a little bit higher because there are a lot of women who actually went through a miscarriage even before they knew that they were actually pregnant. They have done cell sequencing analysis, and they have found that in 60% of spontaneous miscarriages at 12 weeks, these fetuses had a high amount of aneuploid cells. So aneuploidy is, it has to be understood as the gain or losses of chromosomes after cell division. So basically, when we have a cell that divides and it generates two daughter cells, we need to have the same amount of chromosomes in both of the cells. When for any reason there is a lagging of a chromosome or one chromosome that didn't make it to connect to the microtubules and to the spindle, we end up with a cell that might have one extra chromosome and the other one a loss of this one. So we know, for example, Down syndrome, where there is a trisomy of chromosome 21, that this is compatible with life, but in some other cases, when the numbers are higher of these aneuploidies, then this is not compatible with life anymore, and then it's when we have a miscarriage. So as I said, this can occur during the cell divisions, and we have different, like multiple time points where this can occur in the first trimester, so during these first 12 weeks. But the reality is that these aneuploidies usually occur at the very beginning of the embryo development. Actually, they occur in the meiotic divisions in the oocyte, the meiotic divisions of the sperm, and in the first mitotic division of the zygote. If we now take a look to this graph here, what you can observe is that the meiotic divisions, the origin of the aneuploidy in these meiotic divisions is actually quite low. It's lower than the 20%. However, what it remains a little bit higher, and it doesn't change over, if we consider the maternal age, is the mitotic origin of aneuploidy. That this happens in, as I said, in the first zygotic division. So what makes this first zygotic division more error prone? Is it like unique somehow, or is it different? Well, it actually is. And the thing is that we have for the first time the centrosome participation. So the centrioles are incorporated to the oocyte by the sperm, at the moment of the fertilization. And they move to this area here that it's the pronuclear interface between the two maternal and paternal genomes. And then they need to duplicate, and then at the end, there is a symmetrical cell division. This occurs in the human zygote, that we have the introduction of these centrioles in the zygote, but it also occurs, if, for example, in bovine or in the horse. So that's why we decided to use these two models for our experiments. So basically, the aim of my project is to study the role of the centrosomes in the first zygotic division. In order to do this, what I will show you later is like the abnormalities we found in terms of the location of the centrosomes during the mitosis, their contribution to the spindle formation, and also at the moment of the cytokinesis, and how all this is related with an euploid. So in first place, the different functions of the centrosome, we have that they act at the beginning in the mitosis, in the microtubule nucleation and chromosome clustering. So in the case of the oocyte, the way that this happens, because we don't have these centrioles, is that the nucleation occurs from the chromosome. In case of the embryo, so in later stages, what we see is that it's more predominantly happening from the centrioles. In a similar way, when we're talking about the spindle stability and organization, what we see is that in the oocyte, as there is lack of the centrioles, the spindle is a little bit more unstable. And usually you see even multipolar spindles at the beginning before they form the bipolar spindle. In the case of the embryo, it's usually much more stable, and also this whole process occurs more rapidly. So what we have in the zygote is basically kind of like an intermediate stage. And what we're trying to see is what kind of mechanism it uses. However, in terms of the cytokinesis, in the meiosis, we have a very asymmetrical cell division, which is the formation of the polar body, which has a very small amount of the cytoplasm. But in case of the mitotic division of the zygote, now it has to go into a very symmetrical cell division. In terms of the centrosome morphology, it is also unique in this first zygotic division because we have the centrioles that come from the neck of the sperm that they look a little bit different from the centrioles that we will find in a somatic cell or in later stages of the embryo. So we have the proximal centriole that looks, as you can see here in the cryo-EM images, more similar to the ones we have in somatic cells. However, we have this second centriole, that is the distal centriole, that looks different, and it has like this kind of dense fibers. And the activity of this distant centriole, it is poorly understood. However, I'm going to show you here a video. This is a zygote with these two kind of centrioles, and in green you're going to see the microtubules. This is a live imaging experiment, and in magenta you're going to see the chromosomes. So you can see that at the beginning, what we have is that this centrosome here, this centriole here, it's not really engaged to the spindle. Later on, it does, and it forms a proper spindle, and then it goes into telophase, anaphase, telophase, sorry, and then the two cells. In later stages, already in a two-cell stage, what we see is that the centrosomes are more active. And from the very beginning, we have one here, and the second one, they form part of this spindle, and the whole process is much quicker. So in terms of the microtubule nucleation and chromosome clustering, as I mentioned before, in the case of the oocyte, the mechanism that it uses is that the microtubules are nucleated from the chromosomes. And in the case of the embryo, it happens from the centriole. So actually what we saw, and here, this is also like a live imaging experiment, and I just showing you some snapshots of the videos we got. What you can see in the first example is that the paternal pronucleus made it to have the two centrosomes next to it. So you have one here and the other one here. However, in the case of the maternal pronucleus, what you see is that they are capable of nucleating the microtubules on their own, coming from the chromosomes, without the need of having the centrosome. In the second example that you see, we have one centriole that it's not part of the microtubule nucleation at the very beginning. And even this one that it's quite close to the prenuclei, what we see is that also microtubules can be nucleated from the chromosomes. So in this case, what we're seeing is that it's using the same mechanism as the oocyte, and not really the mechanism that we see later on in the embryo, where the nucleation is only happening or mostly happening from the centrosomes. In terms of the positioning of these centrosomes in the prenuclear interface and how important this is in terms of clustering the chromosomes, in this paper, they have shown that it's very important that they stay in the middle between the two prenuclei, and the fact that sometimes they are not part of this prenuclear interface, what they found is that the chromosomes, they cluster less, and then you end up having issues that some of the chromosomes are not attached to the spindle, and later on you have lagging chromosomes and the formation of micronuclei. And indeed, they see that when there is no centrosome at all here at the middle, there are more segregation errors. In terms of the organization of the spindle poles, we have observed that in some cases, not many, only 5% of the videos that we have studied, we see that the centrosomes are actually misaligned. And this means that instead of having one centriole in each of the poles, for example, here we have both of the centrioles in the same pole, as you can see, and they remain like this during the whole mitosis. In this other example, what we see is that one of them is actually in the right pole, and the other one, for some reason, it stays at the end of the metaphase plate. But we didn't see any problems in terms of segregation of the chromosomes later on. What we did observe is that when we have a misaligned centrosome, so a centrosome that it's not really in the middle of the spindle, but it's in one of the sides, and then we have another centriole that is not engaged to the mitotic spindle at all, so here, this one. In this case, I'm showing the microtubules in green and the chromosomes in red. What happens is that it kind of forms two spindles, separate spindles, and then at the end, this lagging chromosome forms a micronuclei, and we have the formation of two prenuclei instead of only one. And then we have also observed, and this is something that we also observed in equine ICSI zygotes and very remotely cases in bovine zygotes. We observed also that there is a fragmentation of the pericentriolar material. So here I show you the peri-centrosome in magenta. We actually we did immunofluorescence for NEDD1, and what we observed were like multiple dots, so meaning that this peri-centrosomal material is actually fragmented, and not only in the area where we should see the centrosomes. And actually, we also did an staining for acetylated tubulin, and we could observe the tail of the sperm still attached to this centriole here, and also in this case. And still like a lot of fragmentation, as I mentioned. Now, in terms of cytokinesis, it is very important where the spindle is formed. In the case of the oocyte, it's usually in one of the corners, so this allows the formation of the polar body. However, in the embryo, it should happen in the middle of the cell, and then we could have two equal cells. What happens we don't have this spindle in the middle is that we end up with an asymmetrical cell division. And you can see here in this video where we have this spindle here in the corner, and indeed at the end of the mitosis, what we could observe is that it generates smaller blastomere here and a much bigger one on the other side. And then another thing that we also observed sometimes is that there is no engagement of the centrosomes to the mitotic spindle. And this I showed you before, but the funny thing here is that it also has a function in terms of the cytokinesis. When this centrosome is way too far from the spindle, what happens is that it's capable of forming a third cell on its own. And then in this third cell, we only have the centriole, but we don't have any chromosomes. So you can see it here when we rotate it, you see that there is only one blastome, like a third blastome that is completely empty, and then the two others. So this is kind of like an idea when embryologists you see a direct cleavage, this could be the reasoning why. So now to finish and to sum up a little bit, the 3 take-home messages that kind of like sum up this presentation. In the first place, that the lack of centrosome activity does not prevent the formation of the mitotic spindle because it still uses the mechanism of the oocyte to nucleate microtubules from the chromosomes, but actually worsens the chromosome clustering. Then that the non-engagement of the centrosomes to the mitotic spindle or misaligned centrosomes, they actually increase the possibility of chromosome segregation errors. And then lastly, that the malpositioning of the centriole can actually give rise to these direct cleavage events. And with this, I would like to thank you all for your attention, and I hope you enjoyed the presentation. Many thanks.
Thank you so much, Ainhoa. It was a wonderful presentation and very, very beautiful slides.
Thank you.
There's a few questions coming in. I'll just take this opportunity to ask something that occurred to me, and it seems to have occurred to Philip Chan as well. And that is, can you speculate on the causes of your observations? So is it temperature that causes these misalignments? Is it something that's inherent in the oocyte, or is it something else?
That's a good question. We don't really know what it's causing, but my idea is that actually this distal centriole, the one that looks a little bit different from the canonical centrioles, it's not really functional or not as functional as the other one. And that's why we kind of seen like sometimes it's misaligned or it's like not taking part of this spindle. So it could be that it's just because the morphology is different, but I couldn't really say that it's related with temperature or the way we are maturing our oocytes or the way we culture our embryos. But we want to like do further investigation on this and see like what is the activity of these centrioles and if there is any way to select for the really good sperm that has the nicest centrioles. But yeah.
That'll be an interesting new study to try and figure out how to see those and then still be able to use them.
Yeah.
Thank you. This is obviously a conversation that we're going to be continuing after the session. So
Great.
Thank you for now.
Many thanks.
Great. And now it's my honour to introduce Professor Anja Pinborg. She's a medical doctor and expert in clinical reproductive medicine and was appointed professor at the University of Copenhagen in 2013. Since 2018, she's been professor and medical director at the fertility department at Rigshospitalet. She's published 270 original papers and authored 9 book chapters and has an H-index of 52 on Scopus, 51 on Web of Science and 61 on Google Scholar. She's a past member of the ESHRE Executive Committee from 2017 to 21 and of the International Committee of Medical Journal Editors from 2015 to 21. And she's a former president of the Nordic Fertility Society from 2011 to 2015. It's now my great pleasure to invite her to present her paper, FET Offspring Health, the most important invited lecture at Alpha.
Thank you, Sharon, for this kind introduction, and thank you so much to the Alpha Conference and editorial board for inviting me to give it. And I will try to share my screen now. Yes, my presentation today is entitled Frozen Embryo Transfer, Health of the Offspring. And here are shortly my disclosures. The setting in the Nordic countries is that 3 to 6% of the children born in the Nordic countries are conceived by assisted reproductive technologies. So a very huge proportion are coming to the world after assisted reproductive technologies. We are still only performing ICSI by indication when it's male factor infertility or fertilization failure. So 40 to 50% of the cycles are ICSI. However, during the latest years, we have seen, and it's even higher now, 30 to 40%, and it's above 50% of the children now that are born after cryopreservation of embryos. So a very huge proportion. But that has also led to the fact that we now have very few multiple pregnancies, lower than 4% in each of the 4 Nordic countries. So in the beginning, we were looking at the neonatal outcomes after frozen embryo transfer, and the results were very reassuring indeed. We saw that in this retrospective register study from the CONATUS cohort. CONATUS is a committee of Nordic art and safety that since 2008 have gathered all the Nordic children born after assisted reproductive technologies, and due to the register recording, we are able to compare them with the full background populations in the Nordic countries. We looked at the neonatal outcomes after frozen embryo transfer, and reassuringly we found that preterm birth and low birth weight and small gestational age was lower in the children born after freezing of embryos compared to both fresh embryo transfer and similar to children born after spontaneous and natural conception. However, when looking at other outcomes such as large for gestational age, birth weight above 4.5 kilo, and post-term birth, then we found that the frozen children from frozen embryo transfer had a 2 to 3-fold increased risk, and that was in the singleton population. We also looked at birth weight curves for the children, and here you see the blue curve is the children from frozen embryo transfer, and the red curve is after fresh embryo transfer. And here you see that the mean birth weight of singletons born after frozen embryo transfer are shifted to the right, meaning that the average birth weight is 150 to 200 grams higher in the children from frozen embryo transfer. So something is going on in their interuterine growth potential. This was also confirmed in this nice systematic review by the Maheshwari group, where they showed that 54% increased risk of large for gestational age after frozen embryo transfer compared to fresh embryo transfer, and also a higher risk of a high birth weight of 4 kilos. So increased risk of about these 45%. They also looked at hypertensive disorders of pregnancy in frozen versus fresh embryo cycles. And again, a worrying result that 29% had an increase in the risk of hypertensive disorder of pregnancy in the frozen embryo transfer compared to the fresh embryo transfer. And here we looked at also a sibling cohort in Denmark, where we compared mothers who gave birth after both a single and after birth, the same mother giving birth to both a child after fresh and frozen embryo transfer. And it could be the first or the second child being the frozen and vice versa. And here you see the birth weight curves. If you first look at the natural cycle or the spontaneous, the second child has on average a higher birth weight than the first child. But if we look at this curve here, where the mother gave birth first to a child after fresh and then after frozen, you can see that this curve is much steeper. So unexpectedly, the child after frozen have a higher birth weight compared to the first child. And on the other way around is if the first child are born after frozen and then after fresh, then the mean birth weight curves go down, even when you have the same mother. So we can say that this higher risk of high birth weight in the frozen embryo transfer children are not correlated to the maternal characteristics, but rather to the procedures per se, either the freezing or thawing, or the protocol, how we prepare the endometrium, and I will come back to that. And here we see the first meta-analysis comparing actually obstetric and perinatal outcomes following program compared to natural frozen embryo transfer cycles. And as most of you know, a program cycle is the same as an artificial cycle or a HRT, hormone replacement treatment, frozen embryo transfer cycle. All these names accounting for cycles where we give estradiol in the follicular phase, and to prepare the endometrium for the transfer, we give estradiol and progesterone in the luteal phase. But what is lacking in this artificial cycle or program cycle? That is the corpus luteum. So in these cycles, the woman has not got a corpus luteum, and we are substituting with estradiol and progesterone until 10 weeks of pregnancy. And here you see in this meta-analysis from Busnelli, this year in Human Reproduction Update, they compared the risk of hypertensive disorder of pregnancy in programmed or artificial cycles compared to naturally prepared frozen embryo transfer cycles. And these natural cycles are prepared in the woman's own cycle, where we wait for the follicle to develop until 18 millimeters, and then either we just transfer the egg after 6 days, or we give her an injection with HEG, that is modified frozen embryo transfer cycle, and transfer the embryo 6 or 7 days, or the blastocyst 6 or 7 days after. But here we see that in these artificially prepared cycles, there is a higher risk of almost twofold for hypertensive disorder of pregnancy. And then going a bit more into the details of these results, preeclampsia in this systematic review was increased more than twofold, postpartum hemorrhage was increased 2.5, and hypertensive disorders of pregnancies increased with a 1.9, and these figures were all highly statistically significant. So something is going on in the cycles without a corpus luteum. This is a very nice study from the Blockeel group in Brussels, where Caroline Rowlands compared 436 women born who gave birth in 2010 to 2019. All had a single blastocyst transfer after vitrification, and they all gave birth in the same institution. So they compared 324 natural cycles with 211 artificial cycles. And here they found in the crude figures 12% of preeclampsia in the artificial frozen embryo transfer cycles. So the crude figures 12% even in these singleton pregnancies, and 3.7 in the natural frozen embryo transfer cycles. So the adjusted odds ratio, after they adjusted for oocyte donation cycles, African ethnicity, and also PCOS, was increased almost with a factor 3 in this very homogeneous group of women, all with a single vitrified blastocyst transfer. And some of the explanation must lie in the lack of the corpus luteum in the artificial cycles. And here are some beautifully performed studies from the Fersen-Hünk and Valerie Baker group, where they have looked at the program frozen embryo transfer cycles where there is no corpus luteum. And in these cycles, a very important hormone called relaxin is undetectable. And on the other hand, there are other hormones and increased, such as creatinine, sodium, and total carbon dioxide is higher without a corpus luteum. We know from studies that relaxin is produced in the corpus luteum at a high level, and we know that this relaxin hormone is very important for the maternal, systemic, and renal vasodilation in the pregnancy. So this hormone is taking part in the transformation from the non-pregnant to the pregnant cardiovascular system in the woman, and obviously very important for regulating the blood pressure. it also increases the kidney function. So from these series of studies in hypertension in 2019, it was concluded that there was an increased preeclampsia risk and reduced aortic compliance with in vitro fertilization cycles in the absence of a corpus luteum, and that is the artificially prepared frozen cycles or the program cycles. And also absent or excessive corpus luteum number is associated with altered maternal vascular health in early pregnancy. So both in the absence, but also in those pregnancies with excessive corpus luteum, and that is the stimulated fresh cycles where we have excessive corpus luteum, was associated with changes in the maternal cardiovascular system. So if you look at this woman, Veronica, she is 38 years of age. Her cycle length is 40 to 50 days, which is too long. She is anovulatory then. She has an AMH, which is on the average quite good actually, on 25 picomoles per liter, but she also has a chronic disease, lupus erythematosus, or a rheumatic disease that further increases her risk of preeclampsia or hypertensive disorder of pregnancy. I know what I would do with her. I would never start her in a substitute cycle unless necessary. So this woman we can induce ovulation in this woman, so we should rather go for a stimulated frozen embryo transfer cycles. And these cycles can be stimulated with monoovulation, like we do in insemination cycles, where we give aromatase inhibitors, or we could give clomiphene, or we could give her gonadotrophins. This is a little bit more difficult, but it is absolutely possible to plan her in a cycle where she actually has her own corpus luteum and then afterwards a corpus luteum. And this Anna, she's a healthy woman. She's only 28 years old. She could be actually my daughter. She has a cycling of 40 to 50 days. She's also clearly anovulatory and has a very high AMH. A little bit more difficult to stimulate for monoovulation, and she's healthy and taking no medication. But taking into account that this woman will also have a twofold increased risk of preeclampsia without a corpus luteum pregnancy, I would still go for a stimulated cycle, even though it can be a little bit more difficult. But I think we need to look much more into, as reproductive endocrinologists, much more into also the stimulated cycle actually, to be able to make better pregnancies for the women, also for the full healthy women that are young and maybe at less risk of preeclampsia on forehand. What about the long-term health and growth of the children born following frozen embryo transfer? We have here looked at the academic performance in the children born after frozen embryo transfer when they are 15 to 16 years. And the reason why we can do this on a national basis is that all pupils at the grading the public school and also private school are going through the same national exams, both written and oral exams. And here we look at the singletons born after freezing of embryos and born after fresh embryo transfer, and they ended up with exactly the same mean scores. And also after adjustment for the most important factor, namely the maternal and educational level, they had exactly the similar educational or academic performance in frozen embryo transfer children, which is certainly reassuring. But what about cardiovascular function and metabolism? We know that the children from frozen embryo transfer are at increased risk of being born large for gestational age and with a high birth weight. And we also know that from the studies on the DOHAT hypothesis, the developmental origin of health and disease, that LGA is actually associated with obesity and insulin resistance later in life, and also for the metabolic syndrome. So what are the risks overall or in general for children born after being large for gestational age? And this is not, I want to emphasize on this, this is not made on our children, this is on children born large for gestational age in general. And we did a systematic review and meta-analysis together with our Swedish colleagues, Magnusson and Berg, where we looked at the risk of LGA, or the risk of long-term disease in children born LGH. And we found that in children born LGA, there was certain risk of malignancies in childhood, and that was in particular for leukemia. We also found that in LGA children in general, there was higher risk of several psychiatric disorders, and also for hypertension in childhood, and type 1 and type 2 diabetes. So these risks are of general importance due to the increasing prevalence of high birth weight babies. So what about the long-term health in the frozen embryo transfer children? We have children that are born after frozen embryo transfer that are actually now up in the 20s or 30s, but they are all born after, and bear in mind, of course, embryo transfer and not blastocyst transfer, so they are born after cleavage stage transfers, and most of them are not born after vitrification, but born after slow freeze. Here, a study from Switzerland causing a lot of attention in all the medias in Europe, at least, where they looked, bear in mind, it is a very limited population of 54 children conceived after ART and 43 control subjects. But what they found in this study from Basel was that there was a higher risk of systolic and diastolic increased blood pressure in the children born after ART, but these were not, bear in mind, not after frozen embryo transfers, but after ART in general. They also found in the ART population in general that the flow-mediated dilation were lower in the ART children, and they found that there was higher pulse wave velocity and higher intima media thickness. And what are these factors actually predicting? These factors are actually predicting a cardiovascular system that would be less adapted to stress, to increase blood pressure. So it's good to have a high flow mediated dilation, it's good to have not as high pulse wave velocity, and it's good to have not too thick intima media to actually be able to adapt to stress. So it seemed that this general ART population, there was the cardiovascular system was less prone to adapt to stress in the ART children. Therefore, we decided to do a huge cohort study on 600 children, 200 born after fresh embryo transfer, 200 born after frozen embryo transfer, and 200 born after natural conception, called the HiCART study, Health in Children Born After ART Studies. And the first abstract was published in the ESHA conference some years ago, and now published in Human Reproduction this year, where we looked at 8 to 9 year old singletons conceived after frozen, fresh, and natural conception. And we looked at multivariate linear regression models for ascending aortic distensibility. And again, aortic distensibility is actually a measurement of how good the cardiovascular system is adapting, how well the cardiovascular system can adapt to stress. And in this model, we didn't see any differences in these 8 to 9 year old children in adapting to stress, neither in fresh versus natural conception, in fresh versus natural conception, or in frozen versus fresh embryo transfer. So also the children born freezing did very well according to aortic distensibility, 8 to 9 years of age. So that is definitely reassuring. We also looked at the autonomic nervous function in children born after frozen versus fresh embryo transfer. And that is also, again, variants that are very important for adapting to stress. So it's called physiological provocation maneuvers. And it is active standing, going from lying position to active standing. It's deep breathing, and it's the Valsalva maneuver. And also in all these maneuvers actually telling us how the heart rate is reacting on these physiological provocation maneuvers. And also the children from frozen embryo transfer did very well in these stress procedures, also at 8 to 9 years of age. And this is actually also published at the ESRO conference this year and will be published in 2023. So also reassuring results. We also in the HiCAT study looked at the body mass index in children from frozen embryo transfer at 7 to 10 years of age and found very reassuringly that body mass index in these children was similar in children from frozen and after spontaneous conception, and that was equal in both boys and girls. So here you see the BMI results for the boys, and here the BMI results for the girls, with frozen coming first, fresh and spontaneous conception. So again, very reassuringly, all very close to the average BMI for that age group. But then came the study here from our group, from the Conatus group, where we looked at the risk of cancer in the full Nordic population, also looking into children from frozen embryo transfer. And here are the results. In the general ART population, we didn't see any increased risk of overall cancer in children born from 1982 in Sweden, 1994 in Denmark, and 1990 in Norway and Finland. The risk of cancer was overall 1.08, and that was not statistically significant, even though you can see that there is a slight increased risk in the overall age groups, but this was not statistically significant. And if there is an increased risk, it's at least very, very low with 1.0 hazard ratio. But then when we looked at the frozen embryo transfer children, and again, it's based on 48 cases of cancer in the children from frozen embryo transfer. And I think here you should bear in mind that overall the risk of cancer was not increased in the general ART population. But in the children from frozen embryo transfer, based on these 48 cases, we found an incident rate of 30 in 100,000 children. So still a very low incidence. We also found an adjusted hazard ratio of 1.59 in the children from frozen compared to fresh embryo transfer, and an hazard ratio of 1.65 in the children from frozen compared to natural conception. And here we adjusted both for macrosomia, birth weight, and major birth defect, and this only slightened the association marginally and not but the results was still significantly. And the most prominent cases of cancer here was the leukemia. It was more or less leukemia, or the majority of the cases were due to leukemia. So I think when we look at this study from PLOS Medicine, that has also caused attention. I think it's very important to state that the risk in general in the ART population is not increased, but there might be an increased risk in the children from frozen embryo transfer, and therefore we should also think about when we do frozen embryo transfers, it should be by indication and not freeze-all for all. So how to minimize the risk of the children born after frozen embryo transfer? I would recommend that you use natural or modified natural fed cycles if it's possible. And I think it's possible if we can more cleverly use the natural and modified natural cycles, because they also have a window of implantation. In case of anovulation, I suggest that we use stimulated frozen embryo transfer cycles, and we stimulate with letrozole or low-dose gonadotropin to gain monoovulation and have a corpus luteum. And in egg donation cycle, only use single embryo transfer in the frozen embryo transfer cycles, because there we normally cannot use other than a substituted or programmed frozen embryo transfer cycles. So to conclude, there is a higher risk of large for gestational age, macrosomia, and preeclampsia and postpartum hemorrhage after frozen embryo transfer and programmed frozen embryo transfer compared with fresh embryo transfer or natural cycles seems to be the protocol where we have the highest risks. A similar academic performance was shown in school children born after frozen and fresh embryo transfer. The cardiovascular health of children born after frozen embryo transfer is reassuring at 8 to 9 years of age in the children from frozen embryo transfer, and also BMI seems to be reassuring in these pre-teenage children that we have examined. So thank you very much for your attention.
Thank you so much, Anja, for that very, very informative talk. I wondered if I could ask you whether the data that you have collected in the Nordic countries and you've just presented to us have actually impacted the practice of ART in these countries, and
Yeah.
so how?
Yeah, it has. We have now changed our protocols, so we only use the substituted cycle if it's necessary. So we always start with now doing the frozen embryo transfer cycles in stimulated cycle in case the women are anovulatory. We start with letrozole, and then we might go to gonadotropins. We might also, in anovulatory women, if we have not succeeded with monoovulation, we might go to actually to the substituted cycle. And bear in mind here, the monoovulation is not as important with the frozen cycle as it is with IUI, because you can always say to the couples that they should try to use prevention of pregnancy when we do the transfer of the frozen embryos. So the monoovulation is not as important as it is in the IUI cycles. And further, in the women that are ovulatory, we would always go for a natural cycle or modified natural cycle first. And that we have done for many years actually, and have quite good experience also with using the implantation window in the natural cycle, which are obviously not only 1 day, but can also be shifted towards 6 or 7 days after HEG, or also you can maybe do the trigger on a follicle that are 16, 17, 18, or 19 millimeters. So there is also a window in the natural cycles.
Right. Thank you very much. Again, there are a number of questions for you, and we'll get to, hopefully we'll get to some of them at least during the Q&A. Before we go to Q&A, I'd like to ask Dean Morbeck to present to us a new initiative at Alpha. It's called Alpha Forum, and I think many people here will be interested in hearing about it, because as it's very obvious with the number of questions that come in during this session and also other sessions on I3, people have so many questions about various topics, and currently we don't really have a great platform to air these questions and respond to them and get opinions from experts shared on these platforms. So, Dean, would you tell us about Alpha Forum, please?
Yes, I'd love to. Let me just see. I've got my screen shared appropriately.
That we can see.
Okay, excellent. Well, thank you. Yeah, so it's, Alpha's got a rich history, as we've spoken about, of bringing people together, whether it's online or in person. And so this is something, as Mina just said, that we want to foster further internationally. We are the global Society for Reproductive Scientists. So this page, and you can see the address at the top here, which is all you need to do is maybe somebody could copy and paste that into the chat, because you can go there and sign up for the Alpha Forum, which basically is an area where we can discuss these different topics. I've got categories listed here. And navigating this area, you can see you can look at topics generally or categories, and you can see there's one that I haven't yet looked at in the principles of embryology. It shows up at the top here, and it's actually really a nice discussion going on. I don't know if anybody's seen Jürgen Lieberman's posts on LinkedIn about their results with a really short blastocyst warming protocol, but that's been an area that's been discussed a bit there. So there's also, going into the topics here, you can have the emails come to you daily, weekly, immediately when there's something added. This just shows you, I put a post to show that you can actually make a little icon on your phone just by taking the web page and putting it on your home screen. So it's a quick and easy way to get into it. And then also, obviously, there's the nice mug there from the group. So this is now live, and we anticipate it's going to flourish with lots of good discussion in these different areas. And I'm sure it'll change over time like anything, but this is where we're starting out. So welcome to the new Alpha Forum. And with that, I think I turn it back to Mina to continue with the Q&A.
Yeah, thank you so much. So I'd like to invite everyone now to join on the screen, and we will start the Q&A section. Anyone who has a burning question or wants to go to the Q&A from the audience and pose a question, please go ahead.
Well, congratulations, Anja. If I may just start, there's a very interesting question from Kevin Leandro from San Jose in Costa Rica, asking the potential impact of slow freezing versus vitrification on offspring health. So were all the threads done without trophectoderm biopsy, and as the biopsy introduces a major confounder compared to fresh embryo transfer? So the first question is about slow freezing versus vitrification, and then the trophectoderm biopsy.
Yeah, thank you for this very relevant question. I think Christophe can answer the first question also, because your study actually clearly showed that also only by including vitrified blastocysts, there was an increased risk of threefold of preeclampsia after the freezing procedure. So I don't think it lies in the freezing procedure. It more lies in the preparation of the endometrium, and it might also be some with the freezing procedure, but I don't think it's also seen by vitrification. But it is fully correct that most of the studies performed on the registers are some years ago, and most children are born after the slow freeze procedure and after a cleavage stage transfer. That's fully correct. Regarding the trophectoderm biopsies, all the Nordic children are born without trophectoderm biopsies because pre-implantation genetic testing without any chromosomal disease or without any monogenic disease is not allowed in any of the Nordic countries, only per study protocol, or if it's in a research protocol. So I can say that the results from the Nordic countries are based on embryos that have not been biopsied.
Thank you very much.
I have a question for Alessandra. Alessandra, how did you choose this particular method of assisted hatching, taking 1/3 of the zona off? And technically speaking, how many laser shots did it require? And do you think that this method could actually confound your results?
Okay, thank you for this very interesting question. We decided to breach a large portion of the zona pellucida in order to be sure that the opening was sufficient. Mm-hmm. Yes, and so we decided to breach and not thin.
And then how many laser?
How many pulses? How many pulses? So we breach 1/3 of the blastocysts. So I think also 10 pulses. Yes, because the portion is big compared to other studies in literature. Because in other studies you can find the assisted hatching with a small hole, just one hole, or thinning or breaching, but only small portion. But we decide a very big, very large portion. Yes.
Okay.
I'd like to just follow up on that one, because I'm curious if there's been, in your program, have you looked at implantation rates for blastocysts that are completely hatched? Many programs find that those are lower. So I imagine there's a state where you might get where there's too large of a hole that allows for hatching before the transfer. We don't understand completely why those implant less, but wonder, this didn't implant less, so they're similar, but maybe the benefit, could there be some, both pros and cons to the size?
Oh, yes, thank you, Dean. I didn't present the data, but in our database, we have the data if fully hatched blastocyst is associated with a higher implantation rate compared to not fully hatched blastocyst. But now currently I don't data, but I can and I will perform a sub-analysis in order to understand if the fully hatched blastocyst is associated with a higher implantation rate.
Shall I ask a question, please, for Ainhoa? Have you made any observations relating to oocyte aging? Do you think that this may play a role in the movement of the spindle towards the center of the egg?
So in this case, what we are trying to see, actually checking more like the mitotic division of the zygote, was to kind of like prevent all the maternal age effects. But indeed, we're doing also studies in terms of the meiosis in the oocyte, that they are related with age. We have a few publications where we have worked with horses, and we actually have horses with different ages, and we can see it. But in terms, of course, of the centrioles, this is, I would say, a little bit more related maybe with the sperm and not that much with the age of the oocyte in this case.
Interesting.
Thank you.
Have you made any observations on tripolar spindles? There's a question from the audience about tripolar spindles and when cytokinesis results in 3 cells.
Yeah, that's a good question, because we do see them very often. And I think also many of embryologists here, you also see it maybe like in the time lapse that sometimes they go from 1 cell directly into 3. And the funny thing is that we have observed it most of the times in polyspermic zygotes, that they always like divide into 3. And then it's the case where we see that there's like also chromosomes in this 3rd cell. However, it's also very common that they have this tripolar division, but then when it's not polyspermic at all, and we know that it's only 1 sperm, then we see that this 3rd cell is completely empty. So it's just kind of like a waste of the cytoplasm, but there's no like a few chromosomes or anything. No, actually the blastomere is completely empty, and we only see it in the cases of polyspermic embryos.
Very interesting observations. Sharon, do you have a question you would like to ask?
Well, I'll ask a question to Alessandra. And it's from John Brown Mensah from Ghana. And he is asking, based on your study, is assisted hatching also recommended in fresh embryos?
Okay, our trial is focused on vitrified blastocysts. Okay, regarding the fresh cycle, the American Society for Reproductive Medicine concluded that there is a moderate evidence that live birth rate are not significant different between assisted hatching and not assisted hatching group in fresh cycles. On the other hand, in frozen cycles, they concluded that there is insufficient data until our trial.
So if I may follow that up, I'm sorry, we're bombarding you with questions, but based on your study, have you stopped doing assisted hatching?
Great question. We discussed with my colleagues, and we are deciding to stop doing assisted hatching because probably it's a waste of time for embryologists.
Of course, for your PGT cases, you still have to open the zona, right?
For PG, depending on the technique of the biopsy, because for example, in our case, in our lab, we perform biopsy on day 5 or 6 directly. So
you still have to open the zona.
Yes, and immediately I do biopsy.
Right, right.
Immediately.
But the zona is open, correct? Which really leads us to a question that I think was posed. I think it was Michael Lee who said, or Michael Reed who said, your study does not show significant improvements, but it also appears that assisted hatching for programs that do this as a routine, that there's not a negative impact either.
Oh, yeah, fortunately, yes, we observe no negative impact, fortunately for us, yes.
Good.
Also for obstetric and neonatal complications.
Yeah. Mm-hmm.
But unfortunately, we have no data regarding long-term effects. No? So in the future, we will follow our children born after this trial.
Anja, can I ask you to comment on this comparison that is sometimes made between getting large for gestational age babies versus small for gestational age and low birth weight and very low birth weight children following fresh cycles. Is that a fair comparison? Is there one that is better than the other?
Yeah, that's a difficult question to answer, actually. I don't think you can say that there's one better than the other, but what we can see is that they are diverging from the mean, which is on average 3,500 when you are at term. So there is something going on in the intrauterine growth potential. In the fresh cycle, it might be that there are more corpus lutea, but there is also a great confounder in the maternal characteristics, because you have women with endocrine diseases, women with fibromyomas, women with PCOS as an endocrine disorders, women with endometriosis. And we all know from obstetric or from the literature that these women have an overall increased risk per se of poor obstetric outcomes. So I don't think you can see that small is better or large is better, but we just see that what we do in the lab and with the endometrial preparation protocols, that it alters the birth weight profiles. And I think it is our duty to try to do it as close to the nature as possible. And that is by trying to achieve 1 corpus luteum in the frozen embryo transfer cycle. And that is by not doing freezing when it's not necessary. Freezing is extremely important for lowering the twin and multiple birth rate, which has definitely been the most important factor for the health of the ART children. Previously, 20 years ago, we saw 30 to 40% preterm birth rates, and we saw neonatal intensive care unit admissions in almost 40% of the children because they were all premature. But today we see a very, very healthy population, but we can see that there is something with these without corpus luteum pregnancies. And I think we should try and adapt to that and try to do it in a cycle where we have a corpus luteum, which is definitely, it's definitely a little bit easier now because we're so used to the artificial cycles. But I think we can also, in a wise manner, use the natural cycles better.
Right.
And
Well,
we need
Thank you.
Yeah.
Yeah, that's a very clear message, I think, to all of us and also to the clinicians. But there is a colleague also from the UK who is asking to speculate a bit about the reasons for the birth weight alterations. Is there maybe a role of the cryoprotectant, specific components in the cryoprotectant that could have a reason for this?
It might be because we can see that the risk of preeclampsia is also increased in the pregnancies where we have in the natural cycles. There is a slightly increased risk. So it might be that the cryoprotection also matters. Now we will start a study where we will look at the pregnancies during from the first trimester, and then look at the intrauterine growth and also look at the placentas in different various protocols of frozen embryo transfer cycles, because that is the only way that we can dig into this. And I'm sure that many other groups are also looking into this matter now, trying to find out what is the cause of this increased risk. But the thing is that we can now see that the increased risk of preeclampsia have been shown in so many cohorts worldwide that it's not a phenomenon linked to the Nordic countries or to the European countries. It's seen in many Asian cohorts, and also from the Asian randomized control trials on freeze-all, we can see the increased risk in the freeze-all cycles. So do freeze when it's necessary, do the substituted cycle when it's necessary with the egg donation and so on, but try to do it as close to nature as possible. I think that's the main message.
Thank you.
Thank you so much. This is a wonderful discussion covering different areas, but all very important. We are close to running out of time. So I would like to invite everyone, if you had a question that was not answered, please go to Alpha Forum and pose your question and start a discussion. Why not continue on Alpha Forum? And so now, before we close, I have a few remarks, and I also would like to present to you the questions that you will have an opportunity to answer. And if you get the answers right to all the 4 questions that we are posing, you have a chance to win free membership to Alpha, as well as a free registration to our next meeting, which in a few minutes I will announce the location for. When you answer the questions, they all have to be in the same chat, and all answers to the 4 questions should be correct. Before I do that, let me tell you why you should become an Alpha member. When you do, you support the mission of Alpha, which, as Sharon articulated, is really primarily education. And you get to network with other ART professionals, especially those working in the laboratory. And you get a free subscription to RBMO, which is a fantastic journal, if I may say, and quite costly if you have to subscribe on your own. You get reduced registration fees for all the Alpha meetings. And just as a reminder, the 2023 membership is only 120 euros per year, or 10 euros per month, if you like to think about it that way. I also want to tell you about our forthcoming initiatives. We are on a mission to expand our membership, and not only expand the membership, but increase our international inclusivity. I'd like to invite those participants from all over the world who might feel that they are underrepresented during these discussions and their needs are not considered or met, to contact us. Please write to us, let us know where you come from and what the status of embryology and ART is in your country. We would like to start collaborations and educational programs that will benefit everyone. Speaking of collaborations, we are continuing to collaborate with other societies and other entities on guidelines and consensus building. And I should mention that our most recent collaboration is with ESHRE on a revisiting of the Istanbul Consensus, and we're going to refresh and renew this consensus. We just sent out, both ESHRE and Alpha sent out a survey for practitioners to respond to and to tell us how useful they have found the Istanbul Consensus on embryo evaluation to be. So please, if you have received the invitation, do respond, but only respond to one of the societies that has sent you the message, not both. We are also thinking about having hands-on workshops and targeted symposia. And we are updating the Alpha website and the logo, which brings me to two opportunities to win today, the free membership and the free registration for Alpha 2024. The first is a redesign of the Alpha logo. If you are artistic and you are interested, send us your logo design ideas and win the membership and the free registration. I've listed 3 email addresses here that you can use, either president@alphascientists.org, secretariat@alphascientists.org, or ecmembers@alphascientists.org. Please write to us on any matter, but send us your ideas of a redesigned logo for Alpha, and you will have a chance to win these fantastic prizes. And now to the quiz. This is your other chance to win membership and free registration. Here are the questions. Please again answer the questions in the chat function, and all 4 answers should be in the same chat. The first person who answers all the questions correctly will be the winner. We may or may not be able to announce it here, but let's see how it goes. The first question is, according to Ainhoa, her first name is easier than her last name, and colleagues, during embryo development, which embryonic division is the most error-prone? The second question from Alessandra and Alessandra's presentation: do fully expanded blastocysts benefit from assisted hatching before FET? The third question is, according to data presented by Professor Pinborg, what is the optimal endometrial preparation protocol for FET in anovulatory patients? And the fourth question is, in which country was the first Alpha Congress held? So again, during embryo development, which embryonic division is the most error-prone? Do fully expanded blastocysts benefit from assisted hatching before FET? What is the optimal endometrial preparation protocol for FET in anovulatory patients? And in which country was the first Alpha Congress held? That brings me to our announcement about Alpha 2024 from 30 May to 2 June 2024. It's going to be in Lisbon, Portugal, the beautiful city of Lisbon. We are very excited about organizing this next meeting, and we certainly hope that we will see all of you registering and attending the meeting. So with that, Giles, do we have a winner? I don't know.
Yes, indeed, we have a winner.
Ah,
it should be in your
okay.
chat. It should be in your chat.
No, you can announce it.
Let me see. It is Veronica Sharon. Veronica Sharon. So congratulations.
Where is Veronica from?
Well, she may like to communicate through the chat. The lucky winner. Tell us that.
Well, congratulations, Veronica, Brazil. Fantastic.
Brazil, bravo.
Hey, bravo.
Which goes very nicely with next week's session, but I shall leave you to say more later.
Yes, okay. So we do have to bring the session to a close. I'd like to thank everyone who took part in this webinar. Our wonderful speakers, of course, for their time, a second time around. First, they gave us their time at the Alpha Congress in Seville, and now again here on this platform. We are grateful for your participation. I'd like to thank my co-moderators and executive board members of Alpha, Sharon, Mortimer, Alison, Campbell, Dean Morbeck, Christophe, Blockeel, and we shouldn't forget that the team behind the scenes from I3 does an enormous job of making these webinars go as seamlessly as they do. We are thankful to them. We're thankful to the organizing committee, Jacques Cohen, Peter Nash, Marianne Cervantes, Thomas Elliott, Giles Palmer, and Fran Farley. Thank you so much to our participants for joining us. We hope you have enjoyed the session. Look out for the accompanying podcast that will be published shortly, along with this session. In fact, listen to all the podcasts and rate them when you have a chance. I3's next session is all about ART in South America. That promises to be, again, an informative and a great session. Don't miss it. Thank you again. Thank you all, and we'll see you soon. Bye-bye.